Hello and welcome everyone to today's expert event on recent developments within the Alzheimer field and the potential sector impact of the outcome of Biogen's aducanumab BLA filing. The event is hosted by Vator Securities and AlzeCure Pharma. My name is David Myslinski. I'm a director with Vator Securities, managing investor clients. The stars of the show will be Professor and Chief Physician Henrik Zetterberg with the University of Gothenburg, a world authority in the field, renowned for his research on biomarkers associated to Alzheimer's disease and Parkinson's disease. Martin Jönsson, CEO of AlzeCure Pharma, and Johan Sandin, CSO of AlzeCure Pharma. The relevance of the research in this field cannot be overemphasized. Every 5 seconds, a person is diagnosed with Alzheimer's disease. I believe we all, including myself, have first-hand experience of relatives or friends' relatives that have been affected. The relevance of this field is also stressed by the overwhelming interest for this event in the investor community, far exceeding our expectations. Moving on to today's agenda. Professor Zetterberg will begin the event by providing an overview of cutting-edge science in the CNS area and Alzheimer's space, and comment on Biogen's BLA filing of aducanumab. After that, Martin Jönsson will give a brief introduction and update on AlzeCure Pharma, which will be followed by a deeper dive into the company's Alzstatin platform, presented by Johan Sandin, and how that docks into development in the field. We will conclude the event with a Q&A session. The Q&A session will be moderated by Felicia Rittemar, Healthcare Equity Analyst with Vator Securities. Lastly, it is possible to send questions via the YouTube live chat function, and we will monitor the chat during the presentation for the following Q&A session. Thank you. Professor Zetterberg, over to you.
Thank you very much for having me. It's such an exciting time in this field, so I love to talk on this topic. My presentation is entitled Update on Alzheimer's Disease Research, New Biomarkers and New Treatments. And I will start by just describing Alzheimer's disease for you quickly here. This is a picture of the pathology of the disease. The disease is characterized by amyloid plaques that you see to the left on the picture, and then neurodegeneration, which is basically neuronal and synapse loss, and then tangle formation. These are the three neuropathological criteria for Alzheimer's disease, basically what defines the disease. During recent years, we have developed biomarkers to measure the disease in living people. And then one can conclude actually that the amyloid plaques are the earliest signs of Alzheimer's disease, followed by tangle pathology and neurodegeneration. And I will come back to that, to this during the presentation, and how we can use biomarkers to better monitor these pathological processes. This is the overarching hypothesis on Alzheimer's disease, the so-called amyloid cascade hypothesis. And it has been widely discussed on whether it's relevant or not. But it starts with the protein that builds up the plaques in the brains of Alzheimer's disease. So in the upper part of the panel you see APP, amyloid precursor protein, and that's a natural protein, all of us have it. It is a transmembrane protein sitting in the neurons and in the synapses, and then it's processed, cleaved by enzymes, giving rise to a protein called beta amyloid. And beta amyloid is the protein forming the cores of the plaques in Alzheimer's disease. And all of us make this protein. It is really a natural breakdown product of APP, the precursor protein. No one knows exactly what it does, but it might have something to do with the synaptic pruning. But it's not established completely. Some of us, actually a rather large proportion of us, will have this tendency to develop clumps of the protein. It's 42 amino acids long, and it's self-adhesive and can form these amyloid plaques. The beta 42 aggregation is what many people believe the core of Alzheimer's disease. That gives rise to oligomers, plaques, that will eventually disturb neural function, giving rise to tau pathophysiology. And tau is a protein that builds up it's part of the structure of the axons. in this simplistic model, a beta 42 oligomers or other types of aggregates of the protein will cause tau phosphorylation. Then axons, the neuronal extensions with which neurons communicate, will destabilize, and then eventually the neurons will die. This is a bit criticized, this model, but it's supported by genetics. So there are familial forms of Alzheimer's disease caused by mutations in the enzymes that cleave out beta amyloid from the precursor protein, and there are also mutations in APP, the APP gene. And all of these mutations cause an imbalance in the production. So if you have mutations, you get a little bit more A beta 42 over other forms of beta amyloid that are less aggregation prone. But there is a criticism against this simplistic model. Some people state that it can't be this simple. And instead, this is another type of model that I like better. It shows, one could think about the mutations in APP that cause familial Alzheimer's disease, or the mutations in the enzymes cleaving out A beta from APP. That could give rise. This simplistic model could be relevant. But in sporadic, the common form of Alzheimer's disease, there are most likely many different pathological processes active in parallel. But to the lower left, you see APP is still relevant in this model. The A beta aggregation, definitely. And many researchers think that A beta is a trigger of the disease, but then there might be a lot of other vulnerabilities in us as individuals that will make us more or less likely to deposit beta amyloid or to react to beta amyloid in a bad way, causing the disease. So this is a slide you could look at and think about regarding potential treatment targets, actually. But I still think that A beta plays a very important role in all this. And if one could get rid of the A beta problem, I think one would solve a large part of Alzheimer's disease, at least. That's my view, based on genetics and also the biochemical effects of beta amyloid. Those studies are also a little bit hard you can always criticize them, but there is such a wealth of data supporting that A beta plays a role, and it's not good to have these clumps, at least. Then one could also think we have genetics and we have age. Those are the two main risk factors for sporadic Alzheimer's disease. That's well established. Regarding genetics and sporadic Alzheimer's disease, we have the epsilon 4 allele of an apolipoprotein carrying cholesterol and other substances in the brain. If you have one of these epsilon 4 alleles, then you often get Alzheimer's disease approximately 10 years earlier than if you have none. And if you have two, it's even earlier. It's almost a gene dose effect on when Alzheimer's start. And this is active in sporadic Alzheimer's disease, so it's a major susceptibility gene. Then there are a lot of other risk factors that have been established in the literature regarding lifestyle-related risk factors. Those have often been found in large epidemiological studies, where dementia rather than Alzheimer's disease has been the outcome. And then it's important to know that dementia is a syndrome of brain failure, and the most common cause of dementia is Alzheimer's disease, but there are other diseases also. Alzheimer's disease make up approximately 70% of the dementias. So diabetes mellitus, midlife hypertension, physical inactivity, smoking, and such things have been epidemiologically associated with Alzheimer's disease. But then it's important to know that many of these factors have been studied in tens of thousands of people. And there is so much literature, popular science literature, and also in the daily press with different recommendations on how you can avoid Alzheimer's disease. I will just go through them very quickly here. Eat more fruit and get rid of Alzheimer's disease. If you eat like in Greece, then you also get less dementia. If you eat fast food, you get Alzheimer's disease. If you do a lot of barbecuing, you get Alzheimer's disease. Curry is good for the memory. Chocolate can reduce the risk of dementia. If you drink wine, you can prevent Alzheimer's disease, but don't drink too much. Coffee is good against Alzheimer's disease. Juice reduces the risk of dementia. B vitamins can halt Alzheimer's. If you brush your teeth, you get less Alzheimer's. If you do crosswords, you get less Alzheimer's. If you take a walk, you reduce your risk of getting Alzheimer's drastically in this paper. If you have many kids, you have an increased risk of getting Alzheimer's disease. if you sleep poorly, you get Alzheimer's disease. If you live close to electrical cords, you also get Alzheimer's disease. If you're close to fume, or if you do a lot of if you're close to fireplaces, then you get Alzheimer's disease, and it continues like this. This is a typical Scandinavian thing. If you do sauna, you also reduce the risk of Alzheimer's disease. Noodles can give you Alzheimer's disease if you eat them. Basically, this really shows how weak this scientific field is. So, and I think this shows why we need drugs, because if you combine this with the knowledge that Alzheimer's disease is defined by formation of amyloid clumps in the brain, we can also measure these amyloid clumps. They will appear in the late midlife, approximately 20 years before onset of dementia. ApoE epsilon 4 will determine to a large extent when you will get these clumps. When they start to form, they form in a very steady manner, mostly like a chemical process. It's very much like the prion disease process that is well known to general audiences through its role in mad cow disease. It's a protein misfolding disease that happens in some of us. It's largely dependent on genetics. It's hard to stop. And these other types of small associations with the risk of getting the clinical expression of the disease, most likely, in my opinion, are related to how well you can handle or compensate for the pathology, but eventually it will all break down. And I think this indicates why we need medical research on this disease. It doesn't help to do these small lifestyle changes. It might change the clinical expression with a few years, but it's not enough. Not that it's bad that a good lifestyle will make you more resilient to Alzheimer's disease to some extent, but it really indicates that we need cutting-edge medical research and that we need drugs against this disease. That's my strong opinion on this. And I think all these newspaper clips indicate why we need good research in the field. We can define the beta pathology using biomarkers. This is an amyloid PET scan. The more red, the more amyloid clumps in the brain. And we can also do CSF sampling and measure a lot of different proteins that relate to the Alzheimer's disease pathophysiology. And lately, we have the blood biomarkers, and that is what I will spend the last part of this presentation on, because this has been such an exciting field. You will see my bias here towards my interest, but anyhow, if one uses the biomarkers and study Alzheimer's disease patients, you can actually classify the patients with memory problems according to if they have amyloid as an A criterion, tau as a T criterion, and neurodegeneration as an N criterion. This is called the ATN criteria of Alzheimer's disease. And that is one way of trying to make this field more objective. And if one uses these biomarkers, it's evident that approximately 30% or even more of people who express a phenotype of suspected Alzheimer's disease, they don't have the disease. They have something else, and we are arguing in the field exactly what they have. But hippocampal sclerosis is one name that often comes up. There is it's not a new pathology, but it's a pathology that has been rediscovered to some extent, called TDP43, which is exciting, but we know way too little about it, and we have no good biomarkers for it. But that could potentially explain some of the Alzheimer's disease-like phenotypes that are negative for amyloid and tau. Still, amyloid and tau are the main culprits in Alzheimer's disease, and that's a bit per definition also, but very relevant to continue to study. If one looks at the clinical criteria, if you just try to make a diagnosis in your office of a patient, you will be wrong around 70% of the time if you're a super specialist. If you're not, then you will be wrong more often, I hope. I almost said now. It should be like that. But it's definitely, by clinical criteria, you are not good enough to make a good diagnosis of Alzheimer's disease, at least not in the early stages, perhaps in the late stages. And TDP43 is one pathology we need to work more on in this field, which is an exciting topic for the future, I think. The risk factors for Alzheimer's disease. Again, if you try to subgroup individuals you study according to Alzheimer's disease biomarkers and study if these potential risk factors causes an increased risk of the pathologies, it is like this. I think I moved to this slide. This is a very nice study that came out. And here you will see that none of the risk factors I listed, except for high age and ApoE epsilon 4, are associated with Alzheimer's disease pathologies. So it's something else. I think it's how the brain handles these pathologies that these potential risk factors and lifestyle-related factors might affect. Still, it could be important, but it's not. It still means that we can't get rid of Alzheimer's disease by changing our lifestyle. Well, you can if you die early, but that's not an option. I mean, that's really not what you would want to do. So we all want to age well, and we should, of course, lead healthy life as good as you can. But then we think, I really think that you will need to attack Alzheimer's disease also by drugs. Yeah. So what do we have for treatments now? The currently available drug treatments are symptomatic. You have clear effects on patients, but the disease process continues. And these are the symptomatic drugs that exist. All patents are gone, so this is super cheap to treat your patient with this. Acetylcholinesterase inhibitors, they will help in the neuronal communication, but the disease process continues. And then you have another drug that you often can start to use later in the disease process, which is a partial NMDA receptor antagonist, also symptomatic. But how can we try to attack the underlying disease process? And now you will see that I base this to a large extent on the amyloid cascade. But in the end, I will also show you some other potential drug targets. Here you see the APP molecule to the left here. It sits in the membrane of many cell types, but it's very highly expressed in neurons. This protein will be cleaved by beta secretase. The most important beta secretase is called base 1, and then you have another cleavage occurring inside the membrane by a protein complex called gamma secretase. And then you will get A beta. And that happens naturally, but in young brains and in healthy brains, A beta does something potentially, but then it's removed from the brain rather quickly. The turnover is 10% per hour. But in Alzheimer's disease, the protein will start to form clumps with itself, and then these small clumps grow and become plaques. And you can look in mouse models, and you will see how synapses around such plaques will disappear and try to take turns around the plaques. And eventually, I think in the beginning, the brain will compensate for this, but in the end, it breaks down, and then you get dementia. The A beta toxicity, the mechanisms why A beta might be toxic are debated. So there are proponents of direct effects of A beta on the membrane or on synaptic receptors or something else. And then there is a model where microglia, the cleaning cells of the brain, are mediators of A beta toxicity. Here you see these small clumps. It would then, from this simple cartoon, be natural to try to inhibit base or gamma secretase, and that has been done. So here is an old study on gamma secretase inhibitor treatments in mice, and here you can see that it works. You can reduce A beta by treating the mice with gamma secretase inhibitors. It works really well, actually, but in patients it does not work, as all of you know. This is data from an old phase 3 clinical trial where it was actually discovered that if you inhibit gamma secretase, you can actually reduce A beta, but you get side effects even, and some of those side effects were pretty significant. Gamma secretase cleaves not only APP but also other molecules, and I will come back to this because it's really important. BASE, the enzyme cleaving out, the first enzyme cleaving APP, really a gatekeeper for A beta production. You can also inhibit BASE. It's a bit difficult to inhibit it, but there are many molecules that can do it now. And again, the biomarkers respond to BASE inhibition in a good way. So BASE inhibition reduces beta amyloid and soluble APP beta. So it works, and it's been shown in mice, in humans, and in also in larger studies. But then this does not have any effect on cognitive deterioration over time. And now we also know that so the BASE inhibitors are rather potent. And the doses given reduced beta amyloid by 60, 70, and almost 90% in a study. So it's a strong hit on base 1 that really reduced beta amyloid. But then last year and the year before last year, it was found that there were also cognitive side effects by by not that the side effects were obvious, so that the physicians involved in the trials could see on their patients that they were deteriorating, but group level effects were quite clear. So currently there is this lack of clinical effect or even side effect, among other than cognitive worsening. This has raised concerns regarding gamma and beta secretase inhibitors as drug targets. And I am so sad about this, because I really think that there is a risk that we might throw away these drug candidates just by these negative effects. One should remember that inhibiting gamma secretase might not have been the best way to do it, and also because there are other substrates, and gamma secretase had such an interesting way of cutting APP and the other substrates. And base 1 inhibition by 70 to 90%, that was probably too much, but perhaps a low dose base inhibitor could prevent the amyloid cascade to happen in people at increased risk of Alzheimer's disease. In my view, these two drug targets still have the potential of being targets for primary prevention of the amyloid cascade. This also means that you would have to treat individuals before they start to deposit beta amyloid. And then you have to start treating healthy middle-aged people who might or might not get Alzheimer's in 20 to 30 years' time. So the drugs have to be super safe. But if one would conclude on the base 1 and gamma secretase enzymes as potential therapeutic trials, gamma secretase there is a potential to modulate gamma secretase function. Instead of inhibiting it, probably affecting many different gamma secretase substrates, you could modulate the way gamma secretase cleaves its substrates. And here these are details, but gamma secretase cleaves sequentially on the APP molecule and probably other substrates also. And then it cleaves at the 42nd amino acid, giving rise to A beta 42, which is very sticky, but it can cleave 40, 39, 38, 37, and those are much more water soluble. And if one could modulate gamma secretase so that you get a little bit more of the water soluble A beta fragments than the longer sticky ones, I think that could be a wonderful treatment. And potentially that could still make gamma secretase work, turning over all other enzymes, and potentially that could be without any side effects. This is speculation from my side, but definitely, according to me, a topic worthy of further investigation. You will hear a little bit more about this later during this in this session. Base 1, I think that if you inhibit base 1 with 30% instead of 60, 70, 80%, perhaps that could be one way of delaying the amyloid cascade in a way that could be of clinical benefit to people. Again, it has to be safe, and then you lower the substrate that will eventually aggregate, and then by that you could potentially make the amyloid cascade happen 10, 15, 20 years later in an individual's life, just by pure chemistry, because the amyloid aggregation cascade is dependent on concentration of the molecule that is about to aggregate. Some other things also, but still. So I would really like to emphasize that I don't think we should stop this research, even though the published data look a little bit but I think there are reasons for why they don't work. That's my short comment on this, and happy to discuss more on it. The base inhibition has support from genetics also, because we have the Icelandic mutation in the APP gene lowering A beta production by approximately 20-30%, and then you are protected from getting Alzheimer's disease. So then we have also the immunotherapies against Alzheimer's disease. There are many different approaches to use antibodies against beta amyloid. Give those antibodies, either you could immunize actively against beta amyloid. That has been tried, but then there were some side effects with brain inflammation. There is still ongoing research on this, but one other way of doing it is to give monoclonal antibodies produced outside the body, give them at high concentration in an intravenous infusion, perhaps once a month or every second month, and then make these antibodies they will cross the blood-brain barrier to some extent, and then bind to the pathologies and help preventing the pathologies from accumulating. Or even reduce the pathologies by potentially activating microglia or by other means dissolving the amyloid plaques. This is one drug candidate called bapineuzumab, where there was clear data that the antibody could reduce amyloid pathology quite clearly. Eventually it was not successful, but the biomarkers moved in the right direction. And one could actually, and then there was some confusion in regards to whether the lack of clinical benefit was a big downside or not, but perhaps also the drug was given a bit too short. And then some years ago, this study was published, which really became an important study, and this is aducanumab. Early phase data showing an amyloid plaque reduction. And then also there was a stabilization of memory impairment, so that the cognitive deterioration was less active. And now you all know that this drug, it had a little bit of a peculiar history, because it was deemed to be a futility analysis indicated that it would not work. But eventually, when all data was in, it was clear that in phase 3 there was one positive phase 3 study, and then another study that was borderline significant. And again, the highest doses really indicated that the drug could get into the brain, remove plaques, and stabilize cognition. And that is really the basis here for why this drug is now being pursued, and Biogen has submitted an FDA application. And this date is super exciting, because here we will know if the FDA will give an approval or not in the US to this. So we are all waiting to hear this. But the data really indicate that removing amyloid could actually help stabilizing the cognitive deterioration over time. And again, the biomarkers have helped us to interpret these trials, because not only amyloid is reduced, but the neuronal injury markers and tau phosphorylation, which are inherent components of Alzheimer's disease, and actually fit into the amyloid cascade hypothesis also, that amyloid causes tau dysfunction, which eventually might lead to dementia. Those markers are also getting more normal in these studies that we have seen data from. And we have also data from BioArctic with another antibody called BAN2401, where we also see the same effects on the biomarkers. So it seems to be safe, and the injury markers in response to the antibody that reduces amyloid in the brain, the injury markers are also normalized. Now there is an ongoing phase 3 trial on this particular antibody. And there are many other anti-A-beta trials that are ongoing now. And if one would summarize the results, I think most of these studies suggest that the antibodies can affect amyloid pathology. Most of them also have a signal on the injury markers. The cognitive data are still a little bit shaky in many of these studies, but again, I have a little bit of a bias as a biomarker researcher, but when tau goes down, neurofilament light, which is an injury or neurodegeneration marker, is more normal, then I feel then I feel hopeful that this eventually will translate into a clinical benefit. Which is then actually what was seen in one of the phase 3 trials for aducanumab. Promising results on biomarkers, less clear on cognition, but I'm hopeful. That's the summary here. And then I have this very busy slide, and it's impossible to go through it, but this is a heroic summary of the ongoing drug trials against Alzheimer's disease, just published by Jeff Cummings and his team. Basically what they did was they have gone through the clinicaltrials.org website and listed all these different molecules. And regarding here, they also detailed whether they are in early phase or in phase 3. This is a zoom in from the same paper on the currently ongoing phase 3 clinical trials. And you see that amyloid is the largest part of those disease-modifying trials, amyloid as a drug target. Tau is a rather small part in this phase 3, but there are many other tau targeting compounds that are being tested. And then there are trials on synaptic plasticity, neuroprotection, some on inflammation, given new reawakened interest in microglia as a potential mediator of amyloid toxicity. So it's a very active research field with, as you all know, a lot of negative news regarding failed trials, but also we have a situation now with a lot of ongoing trials with a lot of biomarker support and a very engaged patient and relative population, and also clinicians who are interested in this disease. So it's a wonderful time to work in this field, and also very exciting to see what happens now in March. And I could just say that, I mean, I really hope that in March we will have some kind of approval in the US of aducanumab. And then my hope is that this will unlock this field, and we will get much more breakthroughs. Not the least with the help of that we now have better biomarkers to know what the drugs more exactly do in the brain. Now I will sum up the biomarker field here. So this is a busy slide with a lot of different candidate biomarkers for different components in the brain. And to the right you see the synapses with which the neurons communicate. There are lots of synaptic markers we can measure. Tau is indicated a little bit more to the left on the screen. Tau is an intraaxonal protein that stabilizes the axons. If tau gets phosphorylated, it will let loose of the skeleton in the axons, and then that axon will fall apart, basically. So that's why this idea with amyloid inducing tau phosphorylation and synaptic and axonal degeneration is a hypothesis many of us believe in. Neurofilament light is another axonal injury marker, which is general to all neurodegenerative diseases. And then we have phosphorylated tau that somehow reflects the tangle pathology, at least the processes that lead to tangle pathology in Alzheimer's disease. And then there are many other proteins we can measure that address different aspects of neurodegeneration, and also some of the other neurodegenerative diseases. In the lower right, I list my two favorite research topics, which are so difficult, and that's alpha-synuclein, where we lack good biomarkers for the Lewy body pathology that is typical of Parkinson's disease, but also dementia with Lewy bodies. And then this other elusive molecule called TDP-43, which forms neuronal inclusions and are present in many people with neurodegenerative diseases and also in the aging brain, but to a large extent in frontotemporal dementias and ALS. No good biomarkers there yet, but a lot of research ongoing, so it's a field to monitor, I think. With the biomarkers, you can classify a living person according to whether they have amyloid, which is A, tau, T, and neurodegeneration N, the ATN criteria. And we have done this in either with PET imaging before, but that's expensive and requires also very expensive equipment. And then we have the possibility of doing it in cerebrospinal fluid samples, and then you have to do a lumbar puncture. In Sweden and a large part of Scandinavian Europe, lumbar punctures are not a big deal. We like to do it, and it has minimal side effects. But it's still regarded as a little bit invasive. I don't really agree on that term, but anyhow. So we have been working on trying to translate the cerebrospinal fluid ATN biomarkers into blood tests, and that is what I will spend the last part of the presentation on. Amyloid can be measured in plasma, but historically the studies published showed it was a mess. You could see studies with increased levels, decreased levels, no change in beta amyloid if you haven't had Alzheimer's disease. So no clear results at all. But in 2017, Randy Bateman's team in St. Louis published this paper. They used immunoprecipitation and a nice mass spectrometry technique to measure beta amyloid. And if you look at the y-axis here, you see that they specifically measured long beta amyloid, the 42 amino acid sticky form, and divided that concentration with the shorter, less sticky 40 amino acid long form. So this ratio really is really abnormal in CSF in Alzheimer's disease. That ratio is reduced by 50%. In the blood, in the plasma, they could see this type of reduction here in amyloid positive individuals. You see them in red to the right in the panel. This reduction is less clear than in CSF, but still the group level difference is quite good. And this was a real breakthrough in the field, which many people got excited about. Then the year after, this study was published by, it's a Japanese-Australian collaboration. A similar mass spec-based technology Again, you use plasma, regular blood samples, and then they could see an increase in the beta 40 to 42 ratio. You will see an increase here because they inverted the ratio. They had 42 as the denominator instead of 40, which I don't really like from a molecular standpoint, but you get better normally distributed data if you do this. But basically, the result is the same, although the ratio is inverted. So again, you see an effect in blood if you have amyloid in the brain. Very exciting. And we made another method, also based on mass spectrometry, similar to the earlier ones, and we could see basically the same thing. This is a cohort where we measured plasma beta 42-40 ratio in cognitively normal individuals in a British cohort where they had or did not have amyloid in their brains. And with this blood test, we could identify the amyloid positive individuals with an AUC of around 0.8, which is good. It's not perfect, but it's actually really good and can be used, for example, if you plan a clinical trial to screen for individuals who are likely to have the pathology against which you target your drug. And this type of screening is good. All of you who do clinical trials will know that screening phase is extremely expensive, and you can save a lot of money by doing this blood test pre-screen, and then you can continue the trial just as you would have planned. But you can remove individuals who are very likely to be amyloid negative by a simple blood test. So we think this is a breakthrough in the field. The other breakthrough regards the T criterion, the tau criterion. So we have plasma phospho-tau 181. I didn't really believe in this that much because the total tau assays were so poor. The ones we had developed for plasma, they really didn't tell much about Alzheimer's disease. But here are data on plasma phospho-tau from a Japanese group where they actually saw this type of group difference. You see to the lower left here, a clear difference between alien controls, correlation, but the dashed line to the left indicates also that the method was not perfect in regards to analytical sensitivity, so more work was needed. Many samples were unmeasurable, but still promising. And this is a study published by Michel Mielke et al. Together with Jeff Dage at Lilly Research Laboratories. They used mesoscale discovery as the platform. That's an immunoassay platform with electrochemiluminescence detection, and they could measure plasma beta like this, and you start to see group differences that look interesting. Total tau to the right doesn't work that well. It's exactly our experience. But phospho-tau to the left starts to look interesting. Then we have this study which is based on the same technology. And here you start to see, it's a bit refined, but you start now to see group level differences that look good. You see the AD, the Alzheimer's disease group in plasma phospho-tau really stands out as compared to the other research groups. And you also start to see nice correlations between amyloid PET and tau PET with plasma phospho-tau. Amyloid PET correlation might seem a bit counterintuitive because we now measure phospho-tau as a tau biomarker, one could regard it. But I think it is like this. Amyloid builds up in the brain in Alzheimer's disease. That will almost immediately result in tau phosphorylation. And that happens before tangle formation. So I think that phospho-tau works almost like a sensor of amyloid. That is how I think this works. And that is how I would like to interpret this marker in clinical trials. If you have a drug that does something to the amyloid process in the brain, to me that should lower the plasma phospho-tau level as a sign that the neurons do better. These neurons are still alive, that has been shown. The neurons that secrete tau and phosphorylate tau, they are still alive, and potentially then they should be possible to rescue by a treatment. This idea that they come out from neurons that are dying and falling apart, that idea has more or less been disproven by the St. Louis team who measured tau turnover with a special labeling technique where they could see that the tau secretion from neurons in response to amyloid was an active process. So that's very exciting, I think. Here another study published back to back with the previous one, showing a correlation between Braak staging of tau pathology in the brain using tau PET imaging and plasma phospho-tau. So you see that already at Braak stages where there is not much tau pathology, if there is amyloid, phospho-tau increases, but then it's a further stepwise increase in phospho-tau levels. So it sort of monitors both amyloid and tau. To the right, you see this amyloid reaction. A-beta positive individuals have higher phospho-tau levels than A-beta negative individuals. We also developed an assay which is on the Simoa platform. So it's a sensitive assay, really reacting to phosphorylated tau and not total tau. This was the pilot study, a clear difference between 80 patients and controls, a correlation between CSF and plasma phospho-tau levels. Here are validation studies showing essentially the same result, and you can measure in both plasma or serums. It's a really robust marker. And this is data together with the McGill team. We are collaborating with the triad cohort with clear group differences. With actually, I mean, it's a blood test that really, it looks like clinical chemistry tests for other diseases. This is another validation cohort. And if you look, the most exciting part of this graph, I think, is the AD group. If you look in the A panel, the AD group in red, and compare that to the FTD group, which is, I mean, this is a memory clinic situation. You have a patient where you think that the memory is failing and there might be some depressive symptoms. Is this depression, frontotemporal dementia, or Alzheimer's disease? And you can differentiate between these two diseases with a blood test with 100% accuracy. So to me, this is starting to look good and like tests that can be used in clinical practice in a good way. And just again, by taking a regular blood sample. There are correlations with tau PET. I won't spend time on this. There's also correlation with amyloid PET. As I explained, phospho-tau is also an amyloid sensor. This is in familial Alzheimer's disease. People who carry a mutation that will give them Alzheimer's disease. Non-carriers are to the left. Pre-symptomatic carriers are in the middle, and you see they start to increase, and then symptomatic carriers are to the right. There are a few non-carriers that have high levels, and we are working on trying to understand that a bit better. This is what it looks like in regards to group level data. Patients with mutations, they start to deviate from individuals with mutations start to deviate from individuals without a mutation approximately 10 years before clinical onset. So it also gives, a blood test gives a window of opportunity to identify people at very early stages. This is data from the cohort I mentioned previously regarding amyloid pathology and the blood amyloid test. This is plasma phospho-tau to detect preclinical amyloid pathology, and it works. It's not, again, not perfect, but an area under the curve of 0.72 for a regular plasma phospho-tau test in people who are cognitively normal, but with amyloid. This is in Down syndrome. With Down, you have a triplication of the APP gene, and it's very common that Down individuals get Alzheimer's disease pathology when they age. And you see that there is a reaction here also. So in the right part of the panel, you see the Down individuals with dementia. This is an autopsy study. And I can basically state here that 8 years before, if you have blood samples 8 years before the autopsy, in the red you see the Alzheimer's disease group with elevated levels. And it correlates with Braak staging, just as we have seen with tau PET imaging, and it's specific to Alzheimer's disease. The other neurodegenerative diseases do not have this increase. Plasma phospho-tau 217 is another very hot phosphoform. It shows really nice results and performs better than phospho-tau 181. But if one would pick one could say that the data from the JAMA paper here favors 217 over 181. But one can also say that if you look at autopsy, the 181 biomarker is better than 217. So more head-to-head studies are needed. Regarding neurodegeneration, neurofilament light works. It's a Simoa-based assay. We can see that it correlates with CSF. We can also see that it works exactly like phospho-tau in familial Alzheimer's disease. Here you see the data that people with familial Alzheimer's disease, the red ones here, there are 2 samples here. They shoot off in their plasma neurofilament light levels when they are about to start to get symptoms. And it works also in sporadic Alzheimer's disease, as I show here. This is ADNI data. This is another familial cohort. I won't spend time on this now, I think. This is what happens in a treatable CNS disease. This is multiple sclerosis. Before and after treatment with an effective drug, you can normalize the biomarker. And if I then would conclude here, we have the promising results for blood A beta as a plaque pathology marker, the A criterion. Less promising results for current assessments for plasma total tau, but very positive results for phospho-tau. And again, phospho-tau 181 and 217 are two potential forms of phospho-tau to measure, and we don't know exactly yet which one to prefer. And I think one could do both now for a while and see what works best. And then there is promising results for neurofilament light as a neurodegeneration marker. And this makes it possible to do ATN classification on blood samples, and hopefully this will be useful in clinical trials. And when we have approved drugs, it's rather easy to envision that this might be used in clinical practice. You see your patient, you measure plasma phospho-tau, potentially beta 42 Vator ratio, neurofilament light, and then you decide on whether this looks like Alzheimer's disease. You might do some additional investigations to be sure. Then you start to prescribe a disease-modifying drugs, potentially targeting amyloid, or if tau is successful, and then you monitor the patient with regular blood tests for the effect of the drug. And if the blood biomarkers do not normalize, you might switch to another drug or increase the dose or play around like that, like we do in general medicine for other diseases. And with that, I would like to conclude. Here is my lab in Gothenburg, in the upper part of the panel, my London lab in the lower part. These are the people who worked diligently on developing the phospho-tau tests in our lab, and this is the McGill team where we did the most of the studies on tau PET and amyloid PET imaging in relation to plasma phospho-tau. And with that, I am done with this presentation. And it's my great pleasure to introduce Martin Jönsson to you, CEO of AlzeCure. Thank you.
Thank you, Henrik.
Please, Martin.
Great presentation. Thank you. Truly. Hello, everybody. My name is Martin Jönsson, and as Henrik said, I'm the CEO of AlzeCure Pharma. I'm happy to be here to give you a company overview as well as a market update, what's happening within the company. But first about AlzeCure. AlzeCure was founded in 2016 out of a research foundation, which was sponsored by many very well-recognized grants as well as from Alzheimer's Fonden. And we are a very experienced team. Many of us have worked in several different companies within drug discovery, research, development, as well as commercialization. And we are based here in Stockholm at Karolinska Institute. And our focus is Alzheimer's disease and pain. And we know, as Henrik said, these are areas of great unmet medical need. And how we are organizing our work, our research work, is based on 3 different platforms. NeuroRestore for symptomatic treatment of cognitive disorders, a very novel platform. And then we have Alzstatin, which is for Alzheimer's disease-modifying treatment. And then we also have a platform for pain, for instance, for neuropathic pain and osteoarthritis. And we are listed on the stock market at Nasdaq First North Premier Growth Market since 2018. We have a market cap of around 360 million Swedish crowns, and we have a cash position of around 150. And our business model as a company is really that we are a research and development company. We develop our projects to early clinical phase and then with the intention to out-license, and through that get income streams from upfront payments, milestone payments, as well as royalties on commercialized products. And this is our pipeline, which we are truly proud of as a company. And as you see, the projects are based on the 3 platforms, NeuroRestore, Alzstatin, and Painless. And as you see as well is that we have projects in several different phases, from research, preclinical, as well as phase 1 studies. And we are intending to start additional clinical trials within, for instance, NeuroRestore for cognitive disorders, focusing on Alzheimer's disease. And this study is fully funded, and we intend to start it in the end of this year. And then we also have a project within our pain platform, Painless platform, for neuropathic pain, and a phase 1 study we intend to start in the end of the year. And also this study is fully funded. But our main focus and really true dedication is for Alzheimer's disease. And we know that this is a devastating disease and it costs society more than oncology and the cardiovascular diseases together. And we also know that the patient population is increasing so much. Over the next 30 years, the patient population will triple up to 150 million people suffering from dementia, and of these, around 70% will have Alzheimer's. And the cost is around, currently, over $1 trillion US dollars, and devastating impacts on people as well as society. So how are we as a company really attacking Alzheimer's? We are attacking it based on the different phases that we see, where we are having the pre-symptomatic Alzheimer, where the disease is building up, as Henrik talked about, and where we have the amyloid beta formations creating toxic fragments and then building up to plaques. And for preventing Alzheimer's, there's currently no available drugs. And then we are working with symptomatic Alzheimer's, when people are having symptoms, memory loss, dementia, and over time also other symptoms based on that the brain isn't working as it should. Here are a couple of drugs, but these drugs are having lower efficacy, severe side effects, and there is truly a need here. So both in these phases there is a huge unmet medical needs, and we are truly focusing on this. And we are focusing on this based on disease-modifying treatment, and we are having the so-called Alzstatin platform, novel oral small molecule therapies targeting amyloid plaque formation, making sure or decreasing the formation of the plaques and the toxic amyloid fragments. And then we also are working on symptomatic treatment with the platform NeuroRestore, also novel oral small molecules targeting neural dysfunction. And when it comes to NeuroRestore, of course, Alzheimer is crucial for us, but we need to say as well that we have multiple possible indications with this in this field, because in many diseases there are cognitive dysfunction which come up with the diseases. So for instance, we see the opportunity within brain injury, brain trauma, Parkinson's disease, sleep disorders, which is so huge, and many other situations. Then, of course, we also have our pain platform, Painless as we call it, targeting also their areas of great unmet medical need. And here we have one project which is TrkA NAM, which is a fascinating project. Here we are focusing on osteoarthritis and severe pain conditions. And osteoarthritis, around 300 million people are suffering from this. And over time, many of these patients may get opioids, narcotic-based substances, which we know can create big problems both for the individuals as societies. So here is a great need. But there is also many other conditions or pain conditions where TrkA NAM could be of interest. So very interesting project. And then we also have a project which in neuropathic pain. And neuropathic pain, there is around 25 to 30 million patients suffering from neuropathic pain. And about 50% of these patients do not experience adequate pain relief. And here we have a project, ACD440, a VR1 antagonist, which is in clinical phase, and we are starting an additional clinical phase as I showed before. Also a very interesting project. So, what are our key milestones for 2020? The milestone has been to initiate a clinical study for NeuroStore ACD856 for Alzheimer's disease, and that we have done. And this study we ended in June, and we got positive data on that study. And now we are heading into our next clinical phase study with NeuroStore ACD856. And then we are working with preclinical efficacy data for our pain project, TREC-ANAM. And also in the end of the year, starting a phase 1b study for our VR1 antagonist project, ACD440 for neuropathic pain. So this is what we are working on to deliver, as we have communicated to the market. And nothing of this has been delayed or affected in any way because of the COVID. So we are very, very happy with that. So why is then AlzeCure Pharma an interesting company? I would say it's because we are truly targeting areas of huge unmet medical need. We have a strong team with a strong track record. We also have platforms with first-in-class property and potential game changers, truly. We also are doing parallel investments in several different candidates with potent follow-up programs, de-risking for the company. And then, of course, these markets where we are aiming to come with drugs for also have multi-billion market opportunities. So with this, thank you for listening in. Look forward to presenting more about the progress we are having. And with this, I would like to hand over to my colleague and dear friend, Johan Sandin, who is our Chief Scientific Officer and one of the founders of AlzeCure Pharma, and he will present our Alzstatin platform. So with that, Johan, please.
Thank you, Martin. So it's my task to follow on, obviously also Henrik's really exciting talk about Alzheimer's disease and the progress that's been made in the field the last years. And I'll be focusing on our disease-modifying platform called Alzstatin. Now, as you heard Martin say, we have two Alzheimer platforms. One is called Alzstatin, is our disease-modifying small molecule therapy platform targeting amyloid production. So it's a gamma secretase modulator targeting earlier stages of the disease. We also have NeuroStore, which is symptomatic therapy, also small molecule therapy targeting neuronal and cognitive dysfunction. So it's a cognitive enhancer targeting later stages of the disease. So we have both of these legs, both disease modifying and symptomatic therapy in our pipeline. Now if we start and focus on Alzstatin. So as you heard Henrik mentioned earlier on, amyloid beta is formed by the sequential cleavage of a precursor protein called amyloid precursor protein, or APP for short. Now this is cleaved by two enzymes called beta secretase and then gamma secretase, and resulting in the release of a small peptide fragment called amyloid beta. And as you heard before, especially the amyloid beta 42 fragment is especially sticky and will over time and with increasing concentrations start to aggregate into different type of amyloid aggregates such as oligomers, protofibrils, fibrils, and eventually these large amyloid plaques that are so characteristic of the disease. As you all also heard is that amyloid aggregates, they are toxic to neurons and make them dysfunctional over time. So this is obviously something we're trying to prevent. So what Alzstatin does, it binds into the gamma secretase and changes the cleavage pattern. So instead forming shorter A beta fragments that are less aggregation prone. Now, this slide shows, and the right-hand side of the slide shows what we saw in the last animation. And as you can see here, you have the APP protein on top being cleaved by first beta secretase and then by gamma secretase, forming these A beta 42 building blocks that will eventually start aggregate and form these different amyloid aggregates. Now, the focus of this project was to find molecules that would target the gamma secretase, but not inhibit the enzyme, but rather change the way that the enzyme cleaves specifically the and forms this peptide. So instead of forming the A beta 42, forms shorter fragments, the A beta 37 and 38 fragments that are not prone to aggregation. In that way, we do not touch the normal physiological function of the enzyme, avoiding a lot of the side effects that are associated with the gamma secretase inhibition. The left-hand side of the slide shows a schematic drawing of Alzheimer's disease progression, where you can see the red curve indicates that A beta amyloid is indeed one of the early pathological features of the disease, while cognitive impairment appears much later, as you heard from Henrik, about 15 to 20 years after you start getting the pathology. And it's therefore important, as you can see also, that the red curve is starting to wear off and plateau at the later stages of the disease. So it's important to go in earlier in the disease to try to deflect this curve and try to prevent downstream effector mechanisms being initiated. So what we've seen with our Alzstatin compounds that are gamma secretase modulators, we've been able to see that yes, these compounds, they do modulate A beta production in human cells. And as you can see here on this graph, with increasing concentrations of the compound shown on the x-axis, we can see a decrease and inhibition of the sticky A beta 42 peptide as well as A beta 40 peptide formed, shown in black and blue. At the same time, in parallel, we rather see then increases in the shorter fragments A beta 37 and 38. Now, obviously, this is also something that we can measure, as we heard from Henrik before, as biomarkers in the clinical studies to show that we have target engagement. We've also been able to show that, yes, with the compounds we can reduce the A beta 42 levels in the brain in vivo in animals, dose dependently. Importantly, we can also see that we do not affect other gamma secretase substrates, the cleavage of other gamma secretase substrates that could be potentially involved in normal cell functioning and play an important role in there. And as you can see here in black and blue, these bars do not differentiate, while a gamma secretase inhibitor shown in green clearly inhibits or changes the cleavage also of the other gamma secretase substrates. So it's a clear differentiation there. Interestingly, we've also done experiments to see how these shorter fragments, A beta 37 and 38, affect A beta 42 driven amyloidosis. And what we can see is actually that these shorter fragments, they do seem to inhibit A beta 42 driven amyloidosis. So if you add that to the medium, you will actually see a decreased amyloidosis in the media. So that's really interesting. So it's actually a kind of a dual effect of these gamma secretase modulators in that perspective. Together with Washington University, we also did some really interesting two-photon imaging studies. So this is a technology where you're able to actually monitor the birth and growth of individual plaques in real time in living matter, so in the brain of animals in this case. So you can actually visualize through a small glass plate in the skull the birth of new plaques being formed. And here you can see on the left-hand side that with vehicle you can see the green arrows pointing to growth of existing plaques, while with compound you can see an arrest in the growth and even a regression of these plaques after a 28-day treatment with a compound. So really interesting data. And it's also important to point out that with gamma secretase modulators, they do display several important characteristics compared to both biologics, antibodies, but as well as the previously tested therapies based on gamma secretase inhibition. Now, GSMs and our compound Alzstatin is intended for oral use, which is a non-invasive, easy to administer for patients and society. So it's a very convenient drug. It reduces the production of toxic A-beta 42, so it reduces the production of these building blocks and thereby preventing the further sequential formation of amyloid aggregates, both oligomers, protofibrils, fibrils, or plaques, whatever is the most toxic species. The mechanism is selective for A-beta, so we specifically target A-beta production. We do not inhibit the enzyme, so we spare normal physiological function, which means that it's a much more safe mechanism. It can produce plaque reduction, as we've shown. And interestingly also, these shorter A-beta peptides being formed are also anti-amyloidogenic, as you saw in the previous slides. And we believe that this mechanism is the most suitable for early treatment of the disease, both given its safety profile but as well as its mechanism of action. And as you can see here on our project pipeline slide, we have currently two candidates, ACD679 and ACD680, in our portfolio. And the project is currently in preclinical stage. And what we've learned, obviously, from the clinical trials in the field is that efficacy and safety is going to be paramount for long-term and chronic treatment, especially if we go in early in what would be visually more seen as healthy patients. Therefore, we're working with multiple candidates to ensure that we have the right and most optimal compound to proceed into the more lengthy and costly clinical studies. We also see that these planned clinical phase 1 studies can assess both tolerability and safety, but we can also assess early efficacy signals based on the biomarkers that we heard Henrik talk about earlier, both looking at levels of A-beta 42 and 40, as well as 37 and 38, which would indicate target engagement, but also these really interesting blood-based biomarkers that we heard Henrik talk about earlier on. So I think this field is certainly moving along in the right direction and for the benefit of our project and the company. So to summarize, so we're working with a mechanism that has strong genetic support. A majority of people with familial Alzheimer's disease actually have a mutation in this enzyme complex, causing a change in the ratio of A-beta 42 to shorter fragments. And what we do with this compound is actually just the opposite. The mechanism that we target, it spares important physiological signaling, which is important and key for safety. So it's a more safe and an inert mechanism. It decreases the production of these A-beta 42 building blocks and reduces all forms of amyloid, subsequent amyloid aggregates, such as oligomers and fibrils, etc. It's a small molecule compound, and we believe this is also very important because it's a non-invasive treatment. It has to be convenient for this patient population, so you don't need to go to the hospital to get injections every other week. So this can be taken at home. It's also a cost-efficient treatment, so it's suitable for this large patient population that we're actually talking about. We heard numbers before on about 35 million people being affected with the disease worldwide. And it also has a strong potential for early preventive use in disease. We talked, Henrik mentioned that earlier, with regard to primary prevention. I believe this would be a really interesting therapy for such use as well. Moreover, this type of therapy could be used together with other disease-modifying therapies, such as antibodies, either as a combination or as a follow-on therapy to antibody therapy. So with that, I thank you for the attention, and we'll turn into the Q&A session.
Okay, so thank you for the presentation. So now we will move forward with a Q&A and panel discussion session, during which questions from the audience will be addressed as we go along. But let's start by addressing the regulatory filing of aducanumab. And what do you think? Quick yes or no? Will it be approved or not?
Yes.
Yes?
Yes.
Yes?
Yes.
Okay. Henrik, can you elaborate on your reasoning?
It's more of a hope for
Okay.
me, but I really think that this is. I think that all the data go in the right direction. I would be very surprised if FDA didn't give this a chance already now. It will be conditional approval, I'm sure, and there will be mechanisms to report back if you would prescribe the drug. But I think that will happen. But and it's also a hope.
I understand. We have a person in the audience wondering if you see any risks with approving aducanumab now on the back of one positive and one negative phase 3 study, instead of doing a robust confirmatory trial before granting.
These two options are both rather good, I would say, but still, I think if one would approve it with this type of demands on reporting back data on biomarker changes, side effects, clinical benefit, then I think that will be like almost like a study, a real-life study. So I think there are minimal risks with approving it.
Okay, and you don't see any risks that that you would approve also onwards false positive.
I think that will be that will get obvious with time.
Yeah. Okay. And Johan, how do you think that an approval would impact the sector?
So I think it would have a major impact on the whole field, actually, because as Henrik alluded to earlier, I think this field needs a breakthrough, and this would provide that type of breakthrough, and also I think provide a step from where research can really leap forward rather actually into this area and get even more interest from pharma industry and from research and focus on this area, because I think, I mean, there's, as we've heard before, there's a lot of genetic evidence indicating amyloid being a really important part of the disease progression and disease pathology. So I think that a therapy that could show that this is actually causing, that reducing A beta can actually also cause a functional improvement, will have a major impact on the whole field, and we can actually also then follow on as the compound gets approved, follow these patients in longer term to see how the effect is over several years. So that will be really interesting. So I see very positive about this.
But and in the event of a potential initial rejection, what are the key factors that still argues for modulating the amyloid beta pathway, still motivating investments into that area of research?
I could just start with briefly mentioning or emphasizing this, that preventing a pathology from happening, then it becomes much cleaner.
Yeah.
And all the third slide I showed with all the different pathways that seem to be active in an Alzheimer's disease brain in later stages, perhaps you could prevent that from happening. I really, when I try to imagine what an antibody does when entering the brain, what does it really do to the plaques? How is microglia affected? How there are so many. It's quite complex. But if one then by making the cascade less likely to happen or making it happen later, perhaps that will make it much easier.
Okay, but in case you had to give up on the beta hypothesis, amyloid beta hypothesis, which of the alternative hypotheses would you lend towards? Viral infection, neuroinflammation, or which? And why?
I think viral infection, those data are exciting, but it's much more research needs to be done there, so I wouldn't believe in that. Inflammation, I think, is downstream of amyloid, and if amyloid falls, then I think, to me, inflammation falls also. I don't think amyloid will fall. That's the thing. But again, it's quite clear that I can't let go of the amyloid cascade hypothesis. Tau, to me, is a difficult drug target, but interesting. but again, in my brain, tau also is linked to amyloid. So I would hope then for, if amyloid falls, I would hope for as another symptomatic treatment together with some lifestyle intervention or so, but that will not be the game changer.
Okay. So what is your take on the tau antibody that failed the phase 2b study?
Yeah, I'm not that surprised, I have to say, because I do not know how the tau antibodies are supposed to work. You sort of throw anti-tau antibodies on the brain, and then exactly what it will do is, to me, pretty much out there. I really think that if you could stabilize tau and make tau, that tau is not phosphorylated, it sits on the microtubuli, stabilizes the axons, that would be a good thing, definitely. I definitely think so. But how the antibody would achieve that, I'm not sure. But I mean, most tau antibody researchers would say now that it will prevent pathological tau from jumping from neuron to neuron, and perhaps they are right. And I hope they're right.
Yeah. Do you see amyloid beta and tau therapies as being complementary in a future?
Yeah, I think they could be complementary. But then I actually, the obvious complementarity to me is an amyloid drug with a good or better symptomatic drug, and perhaps also some inflammation microglia modulator.
And I think that's an important point that Henrik makes, because I think in the field we will see more and more of combination therapies. We're talking about disease pathology evolving over many decades. So it's most likely that you will have better efficacy of certain types of target mechanisms during different stages of the disease. And at some stages you will see a good combinatorial effect or synergistic effect of different types of therapies. So I think that's...
Imagine if you could then use an anti-beta antibody and help extracting beta amyloid or reducing the plaque pathology. And you do that for 1, 2, perhaps 3 years. You do an amyloid PET or a blood test or a CSF test and see that, okay, now the patient actually is treated towards amyloid negativity. Then you add in a low dose of gamma secretase modulator or a low dose of a BACE inhibitor, keeping the patient amyloid negative afterwards. And that type of combination, that would be, to me, wonderful.
So how do you think that the potential of, or the arrival of disease-modifying drugs will impact the commercial potential of symptomatic treatments?
I would say yes, there will always be a need for symptomatic therapies. Now, at least with the amyloid therapies, I think we all agree that we need to move in early on in the disease stage, but at the later stages of the disease, I think there will be a continuous need for symptomatic therapies. Now, for the patients, for most of the patients, actually getting this kind of symptomatic effect and cognitive enhancing effect will, for the affected individual and the relatives, be more profound than if you say, well, we'll change the slope of this curve during 18 months. Of course, that's important for long term, but the immediate effect is much more visual for the patient and the relatives. And I think that will be still a continuous need for symptomatics in the field, especially novel symptomatics in the field. So I think it shouldn't be forgotten in the whole area of disease modification that there will still be a need for symptomatic therapies.
And also, we must not forget the other neurodegenerative dementias.
Yes.
Because I mean, there are lots of people with those diseases, and some of the symptomatic drugs the new symptomatic drugs that are in development in different, they might work to help those individuals. Amyloid is part of dementia with Lewy bodies, that's for sure. In Parkinson's disease dementia, amyloid also seems to play a little bit of a role. But then we have frontotemporal dementia, where there are completely different disease mechanisms in action, and there we need those we need to address by other means.
So over to biomarkers and diagnostic tools, specifically for clinical trials. How is that area developing?
That has really been the last year has been extremely interesting with the phospho-tau markers from different research groups using slightly different methods and very concordant results. And that's when I believe in in a biomarker when if one lab shows fantastic results, when they start to get replicated by other research groups, then I start to think that this is probably right, and that's the case here. So we have, as I mentioned, blood tests. We shouldn't forget about the CSF tests and the PET imaging. The PET imaging also, although I'm not an expert, it gives you anatomic information on where in the brain the pathology sits, and that will be important in the clinical trials when you look at disease progression. The fluid biomarkers have more of a tendency to be binary, but there are also some quantitative information that you can harvest from the fluid biomarkers. I believe that we should continue now working with all biomarkers in parallel for a while, and then eventually develop algorithms where you might actually phase out some of the most expensive or redundant biomarkers. And for clinical trials now, I really believe in this type of pre-screening with a blood test to reduce the number of unnecessary expensive examinations.
And as of today, what is the level of recognition regarding this subject among regulatory agencies?
It's really,
And what is needed to bridge between what is happening in the scientific field towards other industry stakeholders?
The regulatory bodies are very interested in the biomarkers now, and there are many supportive statements from FDA and EMA and other regulatory bodies. PET, of course, has been approved. CSF is on the way. The blood tests are also. What we need to do now is to standardize the assays, to develop global reference standards, so that we measure. The clinical labs can start to measure at the same level. We can use the same reference limits and the same cut points. All that will increase the interpretability of the biomarker results. But that is that's predictable. That is just working together to achieve this, and we know we will get there. Finding the biomarkers is more random and discovery based, but now we have the biomarkers. Now we just need to standardize them. And in clinical chemistry, we have done that for all the other tests that we use in emergency care units, and whenever you visit your physician, there will be blood samples taken.
So it's pretty de-risked then?
There is no risk with this, exactly.
Okay.
That is a very small risk.
Okay. So how are biomarkers implemented in AlzeCure's planning of clinical trials?
Yeah, so as I mentioned earlier on, and I think we heard in Henrik's excellent talk about these new biomarkers being developed, and I think the whole field is actually working in our favor in that perspective. But we do have already now obviously included biomarkers where we can look at target engagement, so looking at 7 and 38 being formed, so similar to what we've seen in the preclinical experiments. We can also assess the type of level of efficacy achieved. So those type of biomarkers. But I think what we heard right now also on the phosphatide biomarkers being some type of marker of amyloid. I think that those types of biomarkers would also be very interesting to include, as well as obviously with kind of neurofilament light or any other type of marker that would indicate some type of functional recovery or functional improvement in neuronal function would be really interesting, besides type of cognitive endpoints, especially in the later phase 2 and phase 3 trials.
With the new plasma beta blood test, we can measure 42, 40, 38, and 37. And when Johan spoke, those were the molecules that their compounds are supposed to modulate. So then, in living humans, one could dose the drug and look what happens in vivo with these ratios. So that's why I think I always feel like the Alzheimer research field is very mature to do these types of studies, because you can monitor your candidate drugs very effectively in regards to mechanism of action, side effects, and beneficial effects using then these types of biomarker readouts. And then we need to link it to cognitive clinical benefit. And that's also why I'm so hopeful that we will get an approval, because if we eventually will have an approval of a drug, we can use information from such data to perhaps eventually get a surrogate marker predicting a clinical benefit, and then I think that will unlock the field.
And why is it difficult today to bridge between the effects on biomarkers to cognition?
Yeah,
Is it difficult to measure, or?
It's a bit difficult to measure, but the problem now is we still do not have the drugs with a clear clinical benefit, and potentially aducanumab can be the first drug that, I mean, one phase 3 trial was really linked those things, but potentially we would see much more data on that soon. And then one could, then the biomarkers I hope to be able to link to a clinical benefit are phospho-tau and neurofilament light. Neurofilament light is a general neurodegeneration marker, but phospho-tau in AD, if one could see a percentage reduction translating into a clinical effect eventually, I mean, that would be so wonderful. I think it will happen.
So what do you think of post-translational modification of amyloid beta as a starting point of aggregation?
Yeah, that's a very good question. So, I mean, the post-translational modifications have been suggested, phosphorylation, glycosylation. In our studies, it's a very small proportion of A beta. The most important post-translational modification is pyroglutamate modified A beta. It's an N-terminal truncation in the other end than the A beta 38, 40 and 42. That modification is linked to Alzheimer's and symptomatic amyloid pathology. It is, if you look at what neurons secrete, very little of that is secreted, almost none. I think it forms in the brain. And then it might form by an enzymatic process or by simple, something called peptide backbone attacks that might make this happen. So I look at that as a marker of toxic amyloid, but I do not think it's an amyloid pathology initiator. I think I almost think it forms in the plaques. So it's a bit downstream, but according to me. But again, if one does research on this, it's an interesting topic, and whether that could help seeding amyloid. That is, there are data indicating that it's very aggregation prone.
So Martin, the field is clearly undergoing an intense knowledge inflection point.
Wonderful.
How do you see that AlzeCure as a publicly traded company is positioned to adapting with emerging science?
We are truly adapting, and I mean, for us to be able to use this additional knowledge that we are getting, working with experts as Henrik and others, and bringing this into our projects, and also when we are looking at the project we are doing, which are on Henrik's wish list, I think it's looking very good, and I mean, we are looking for additional cooperation with different groups who are having great knowledge on this. So I feel we are very, very well positioned.
Yeah, I think that we will conclude the event now. Do you have any final remarks or take-home messages that you would like to convey?
Henrik, please.
I'm simply excited about what's happening now. That's my and I have. I think this will be it's an exciting time in this type of research. That's my general conclusion.
Yeah, I was thinking just to say, that we are really at a really exciting stage right now in research, both in the biomarker field, in the therapy field, and I think. There's a possibility that in the spring that will actually provide a real leap forward in the whole field and more focus on the whole area, which I think it would benefit. So really looking forward to it.
And I think as well we are standing in front of a breakthrough. I think it will mean so much to the field. I think this will lead to so much more investments in this field, which are truly needed. Because if we look at the investments that are done in Alzheimer's disease, it's about 1/20th of oncology. So why don't we have a drug today? I would say we haven't invested enough. We need to invest more. And I think the breakthroughs and the additional knowledge that we now are having are going to make the money come into the research, and then we will also have more drugs. So it's a perfect time, and we are looking forward to do more projects, looking at microglia, for instance. Then we need to see. Yeah.
Well, thank you, Henrik, Martin, Johan, for participating, and thank you to the audience for listening in and asking questions. Thank you.
Thank you very much.
Thank you. Take care. Bye bye.